Thank you, Amy, for the introduction, and thank you to you, Mark, for this great opportunity. Today, I want to talk to you all about new tools for NMR-Metabolite Biomarker discovery, which are Navigators, NUS, and Signal Enhancement. My name is Alessia, and just as a disclaimer, I work at Olaris, which is a private company from which I get both financial support and equity. Today, I want to talk a little bit about what are metabolites and why they're powerful biomarkers, but just as any other awesome thing, they're also challenging. So I want to talk to you a little bit of how we try to overcome that at Olaris using our CEREBRO platform and how you can add metabolite profiling to your research. But the main take-home message that I want to give you guys today is the importance of QA/QC. In fact, we all know that the quality of the data that you put in will determine your results. So if you put garbage data in, your results will be as bad. But if you put shiny, beautiful diamond data in, your results will be equally as great. So what are metabolites? Metabolites are all the molecules that are present in a system or in a biofluid, for example, at a specific time. All the traditional omics sciences give us more of an idea of what could happen, like genomics and transcriptomics, and proteomics gives us an idea of how that happens. But metabolomics gives us the full picture of what's happening in that exact moment, and that is why are so unique and powerful. In fact, just analyzing, for example, your urine, I can get an idea of your sex, of your approximate age, of your diet, of your ethnicity, if you traveled recently and where you traveled, and if you have any health conditions, and also what is the best treatment for that health condition just for you. And that's why it's so fundamental to study the metabolites to understand what's the best diagnosis and what's the best treatment for that specific person. That's why they are so important for precision medicine. If we think about our everyday lives, metabolites are already measured with diagnostics. So we think about going to the doctor and getting, for example, a blood test. What are the things that are measured? You can measure glucose for diabetes. You can measure all the lipoproteins and cholesterol and lipids for your cardiovascular health. There are other metabolites that are also used for treatment, for example. So L-DOPA is used to treat Parkinson's disease, but it's also how they tell that somebody actually has Parkinson. There are more and more tests now that are coming out that use metabolomics for diagnostics, because the altered metabolism is linked to almost every single disease. But why are they so challenging to measure? So they have a very broad chemical diversity. So we can think that they are very polar molecules or polar molecules like lipids. You can have a great dynamic range. There are molecules that are present in your system at femtomolar concentration. There are others that are millimolar concentration, and they're constantly fluctuating. So what you have in your urine in the morning is completely different than what you can get at night, for example. So to take into account all of these things, you probably have to use multiple platforms to get a full picture, because a specific platform can be targeted to a specific class of molecules or can be better for a specific concentration of molecules. In addition, you have to consider what we said at the beginning. So as metabolites give you an idea of everything that's in that system, they are also affected by confounders. So, for example, by BMI, by age, by sex, and all of these things. But in spite of these challenges, at Olaris, we still believe that the metabolome can transform care, and we try to do that. How do we do that? So we use the CEREBRO platform, and I hope somebody gets the reference. We get our samples, we analyze them with both NMR and LCMS because we believe in the power of combining these platforms. We have our feature identification, all our data processing, and then machine learning algorithm that can finally give us a predictive algorithm for a treatment or a disease. But we firmly believe in the link to biology, because everything that comes out of this machine learning has to make sense. If something doesn't make biological sense, it's useless for medicine. In addition, the fundamental part of this is the beginning. As I said, the data that you put in will influence your results. So sample collection and data acquisition will determine the quality of your results. Therefore, it's key to ensure the high quality of your data. And how do you do that? With robust QA/QC, as mentioned, and monitoring every step of the way. So these are the main steps that we'll go through, like from when you get the sample to the data processing before you do all your fancy algorithms. So we can think about biofluid collection. Even here, you can introduce some error and some bias. So for example, what type of biofluid are you using? What type of fluid are you using? Are sample fasted? Are they not? And then we go to metabolite extraction. Are you using any extraction method? Do you use protein precipitation? Do you need to extract the lipid layer? What are the solvents that you use? What are the buffers that you use? We get then to data collection. What is the best platform for your project? Do you need MS? Do you need NMR? If you do NMR, is it 1D enough? Or do you need 2D? What are the best pulse sequences? And then we get to data processing. Is, for example, for NMR, automatic baseline and phase correction enough? Do you need to introduce like uh, some manual phase and baseline correction? So in that case, like you can possibly introduce like a bias from the operator. What is the best normalization? And with that, I can start like a 2-hour discussion on this. Do you need to remove any outliers, and how do you spot them? So each of these phases is capable of introducing a bias and an error. So it's key to make sure that every step of the way is controlled and you can get the best data out of it. So let's start from the beginning. Let's start from biofluid collection. You need to decide what biofluid you're picking. So we saw in a previous study that metabolites from the same patient are found different in different biofluids. And this, for example, is expected from plasma versus urine. But plasma versus serum, some people think, blood is blood, but that's not the case. So as you can see here from this Venn diagram, this data was presented at San Antonio Breast Cancer Conference a couple of years ago. We found that metabolites were different between plasma and serum of the same patient at different time points. So before they started treatment, and at the beginning of treatment, and 15 days after. And this tells you that even if the subject is the same, is at the same time point in treatment, metabolites differ. So it's fundamental to pick one specific biofluid. Moreover, we saw the metabolites from the same patient in the same biofluid are different between different collection tubes. When you go to the doctor and you get your blood drawn, you can see that the tubes have different caps. So what does that mean? Each cap will tell you the type of anticoagulant that is present in the tube. We try to measure the differences in the different tubes, and we saw that there's a high number of metabolites that pass a Kruskal-Wallis nonparametric test, and these are much higher than the number of metabolites that would pass by chance. So this tells you you need to pick a specific tube for your analysis. But that's not always in real life, because sometimes we just get like the scraps from all the other analysis. So, or maybe like you have to combine tubes from different collaborators for a study. So how can you avoid having confounder there? So we believe in the use of a strong fold change filter. So for example, we use 1.5 times the fold change as a threshold, as a cutoff. So just thinking of that, here we get a much lower number of molecules that pass the KW test and the fold change. Even better, you could use a false discovery rate correction for multiple comparison, and in that case, you can see here that no metabolites pass that. So we highly recommend using more and more of these filters for your analysis, and that way you get more reproducible and more accurate results. Then we can go to metabolite extraction. So how do we get the best data from this part of sample prep? Metabolite extraction is different from different labs, so you can choose to do no extraction. Some people do ultrafiltration. In our case, we do methanol chloroform extraction, the Folch method. But then also you have sources of error from the samples itself. Samples can have a different pH or specific gravity. That's particularly the case for urine. You can get different volumes from your collaborators, and also you can get an error from the operator that is handling the samples if you have different people in the lab. So at Olaris, we have developed some molecules that we call Navigators that can be added to monitor all these sources of error. The first one, we call the sample prep navigator, or Navigator 1, is a navigator that can be added at the beginning of sample prep and will tell us if sample prep went right or not. So as you can see here, we have two spectra of urine, and the difference basically is only the presence or absence of the navigator. We also checked, and it does not affect, for example, the sample pH. It can tell us if there's something wrong with our preparation. How does it do that? So we checked, and if we use different operators or the same operator, many different samples, we have a very low CV%, low variation in the samples in its intensity. So we have 3 to 10%, but if something goes wrong with the preparation, for example, we forget to add chloroform so we don't get lipid separation, or we drop like half of our aqueous phase, which, small Eppendorfs, they have to be open, can happen. Then the CV% dramatically increases. So using a threshold of 10%, for example, of the variance, we can know that something maybe was wrong with that sample, and so we flag it, and then we can check later if that sample needs to be removed or not from our analysis. Another thing that varies a lot, especially in urine, as mentioned, is pH. So we've seen in real life, in our samples, human samples, it's extremely varied. In rat samples, for example, even more. We had samples that were pH 5, some samples were pH 8. So how do you monitor that? In a paper that was presented by Mike Riley, this molecule, dimethylcyanylphosphonic acid, was synthesized and presented as a chemical shift indicator of pH. So in fact, its chemical shift changes with pH of the sample. And we saw that it was linear both with the initial and the final pH of our samples. You can see that like when we prepare samples in our way and then add our buffer, the range of pHs in our sample is much, much lower. But still in that case, we can still get a good idea of the pH of the sample using the FDMP. Why is this so important? Because metabolites change so much in relation to pH, even in the small buffer range that our samples are in. Here is a paper that was presented by Bob Powers, by his group, and you can see that all these molecules change their chemical shift both downfield and upfield in relation to pH. And you can see here, like even when the pH changes are small, you can get a good difference in chemical shift. So why is this so important? Because we talked about how it's important to identify molecules and how it's important to correctly compare the same thing in our samples. In this case, for example, we have histidine and we have a spectra from one patient and from another patient. And we have to know if we're comparing the same thing between them. So we can see the signal on the right from histidine, the one marked in green. You can more easily tell that it's the same molecule. But if we go to the signal on the left, the one with the orange asterisk, you can see that the chemical shift difference is great. And so are you sure that this is the same molecule? When you have many samples, that's difficult to tell. So using the navigator, we can have an idea of the pH of the sample and be able to better compare these molecules. You can see here, if we zoom out and use all the samples from the patients, we could tell which ones had a specific final pH. For example, this group had a final pH around 6.8, this group had a final pH around 7.14. And in this way, you can know, for example, that this metabolite is varying this way and with pH. And then you can get a bigger tolerance window just for the specific metabolite, because you know that it's so sensitive to pH. And you also know from the FDMP that these patients were wildly different in their pH. Of course, in an ideal study, you would adjust the pH of each sample, but that's not always feasible because you might get very, very many samples from your collaborator. And in that case, it's not possible to just do everything before sample prep. Moreover, you maybe don't want to introduce any bias, any error. You can introduce cross-contamination of your sample if you measure each pH. So in this case, the use of the FDMP is fundamental and will help you to compare the same molecule for all the patients. Another thing that changes greatly in urine is specific gravity. So this is a usual readout that can give you an idea of the dilution of the sample. It's used for hydration, for example. So if a sample has a very low specific gravity, it means the subject was well hydrated. If it has a high specific gravity, it's in serious dehydration, and it can be in relation to some diseases. And we saw that the same signal to noise, just the same pooled urine diluted to different specific gravity at wildly different number of peaks. So as we've seen that this can actually happen in real life, how can you compare these samples correctly? We saw that using the intensity of metabolites, for example, like creatinine, we can get a good correlation with specific gravity. In that way, knowing that the range of specific gravity in our sample, we can help guiding the cutoff of noise to signal, for example, and normalization of our samples. Then we go to data collection. So how do we get the best data from our NMR? Traditionally, in metabolomics, 1D NMR was used because it can give a direct quantitation. It can still give you structural information. It's highly reproducible. It's high throughput. You can get a spectra in very few minutes, and you have all the information that you need there. But the problem is that there's still a great chemical shift overlap, especially in biofluid where we have a lot of molecules. And in that way, it's almost impossible to get correct metabolite identification for many metabolites. And this is fundamental for the link to biology that we mentioned at the beginning. That is why at Olaris we use 2D NMR, so we use proton carbon, but this is based on the carbon-13 natural abundance. So that means that we need extremely long acquisition times to get a good spectrum. How do we overcome that? We use NUS, so we use non-uniform sampling. We use a Poisson gap sampling schedule. We use 25% NUS, and then we reconstruct it using HMS-IST, and then we get our data. And we saw that using the same time of a uniformly acquired data, we get a lot more peaks with a NUS spectrum. Moreover, we saw that the linearity was perfect for some of our molecules that we tested. We had a low limit of detection, and then we had a low CV%. So the majority of our peaks had a very low variation, and all the small peaks that had a high variation are probably stuff that you don't want to even look at anyway, because they're probably artifacts. And we saw that with a 25% NUS, using a quarter of the time, you can fully recapitulate the full spectrum that is acquired with uniform sampling in plasma. Another thing that we tested to gain a little bit more time and a little bit more signal intensity is using every information about the NMR. So we can use a lower cycle delay but a higher number of scans. We can use PRE agents, paramagnetic relaxation enhancement agents, that can help us accelerate the relaxation of the nuclei in our sample. In fact, using a lower D1 but a higher number of scans, we had a great increase in signal to noise ratio. We got signal enhancement. And the same thing happened, for example, even better when we added gadolinium as a PRE agent. In fact, we saw that the signal to noise ratio increased and then our peak intensity increased. Moreover, we got a lower CV% and a lower limit of detection, so we could actually see more molecule, which is fundamental in metabolomics. In the end, if you get a table like this, you're in a really good spot to do your metabolomics data project. In fact, you can have samples like this one that were flagged because the Navigator 1 intensity was bad, or DSS was not detected, so your sample was probably proteinous. So you should probably discard that sample from your data set because you don't want bad data in. These other samples were flagged. For example, DSS was not detected. The Navigator 1 intensity was weird. So you can go back and check if those samples also need to be removed. In this way, having all this information, you know what you have to remove, what you can keep, and what your pH was, what your specific gravity was. So you can know how to cluster together your peaks, how to normalize your sample, and then you can get the best results out of your data set. So what we've seen today is why metabolites are so powerful as biomarkers, but how are they also challenging to measure? And I talked to you a little bit of how you can mitigate these challenges. So you can use a navigator for sample prep. You can use a pH navigator, a specific gravity navigator. We're also doing some work on a protein contamination navigator, but I didn't have time for that today. I can use NUS and signal enhancement tools to improve like your signal to noise, your limit of detection, and your enable lower variation in your samples. So the take-home message, QA/QC is the most critical step for metabolomics. If you put beautiful, shiny diamond data in, you can get beautiful, shiny diamond results out. And how can you do that? You can collaborate with us. So you can see that we have a great list of partners, and you can be one of them. And I want to thank you all for today for listening to me. And if you have any questions, you're very welcome. And I want to thank my team for all the help with all this work. I want to thank Liz for this opportunity, and all our great friends and SAB and board members and like Bob Powers, Frank, Sven, and Garrett for all their help and support throughout all these projects. So thank you all.